I was like, then I won't put that.
Okay. Slides are now up. Sorry. We can finally start. All right, everybody. Thank you. Welcome back. We've got 2 weeks left. And so the next sort of set of 4 lectures, I'd say are all sort of related to each other. They're all about the brain and how the brain interfaces with its environment. Basically how the motor system works, how sensory systems work, how the brain itself is a computational device, and how its functions are really determined by electrical changes within cells, and how those electrical changes are going to get transmitted down long wires and release chemicals and pass on and trigger new electrical changes in the next set of neurons or the next set of cells, right? This is sort of this image here is the image of a slice of a hippocampus from a mouse called a rainbow mouse that was developed, I think, in around 2007, where they were able to use genetic schemes to randomly label neurons within the hippocampus with a combination of different fluorescent proteins. And by doing this, their hope was to be able to trace the exact circuitry of the hippocampus by following these different colors, and then be able to determine the computational characteristics of the hippocampus, how its function works, just from how the wires lined up. Okay, this is because all of the functions in the brain are determined by the connectivity patterns of the neurons that are sending electrical signals. Any sort of filter, any sort of like process, any program that the brain wants to run is going to be determined by how the connections between individual neurons are formed and how they are maintained, right? And what electrical signals are going to get sent out and how will that be processed. In the end, I'd say actually the rainbow mouse was pretty disappointing, because there's just not enough colors. There's too much overlap here. You weren't able to trace the thing. So it made a ton of pretty pictures, but did not actually solve the hippocampus because we weren't able to trace things that well enough. But new techniques have come out with more sparse labeling that actually have been able to sort of reveal a lot of the secrets of how circuits in the brain can generate computations. Now for these, so this sort of set of 2 lectures, or I guess it's maybe 1.5 lectures, is going to be on cellular neuroscience. So there's different scales in which you can think about the brain, think about neuroscience, right? You can be a molecular neuroscientist, okay, or somebody that is really studying like the actual chemicals, maybe the pharmacology of specific receptors, right? How do those channels work? How do they get modulated? And then, of course, if we put those together into a cell, how's the overall shape of a cell sort of lead to a variety of different computations that you can do? How could you sort of add 2 numbers together with like basically a cell, and what shape would allow that to happen? And then you can expand that into circuits, which is where we are going to wire up multiple neurons together into some sort of computational unit. And then finally, those can be combined into systems. So how a systems neuroscience would be like, how does the visual system work? How does your sense of smell work? How are you able to control your arm? That would be sort of motor systems, right? So there's all these different scales. And then finally, we have the brain, which is sort of the total nervous system scale, which is where you put all the systems together to accomplish the overall function of the brain. So we're going to sort of start at cellular neuroscience, and in part of that process, there will be a lot of discussion of particular molecules. Ion channels are going to play a huge role in cellular neuroscience. And the key concepts I want to really hit here are, 1, how does the structure of a neuron accomplish its function? That is, how is the shape of a neuron, and what chemical molecules it can release, and what receptors that make it sensitive to chemicals that other neurons are releasing? How does that going to accomplish the function of that neuron? What is the function of a neuron? How are we going to take information, and how are we going to take information and move it from one neuron to another neuron? How are we going to transform that information? Are we going to filter it in some way? Are we going to multiply it in some way? How does that work physically? Okay. Then neurons are not the only cell in the brain. In fact, they're only like 10% of the cells in the brain. We actually have a whole bunch of supporting structures, glia, okay, that we were going to touch on briefly. Okay. Then we're going to come back to how does this sort of how does an electrical signal even work, right? In a cell, how do you generate an electrical signal, right? And that is all going to be determined by something known as the membrane potential, which is an electrical difference between the inside and outside of the cell. Most cells in the body have an electrical potential. Usually, the inside of a cell is more negative than the outside. Okay, but usually that membrane potential doesn't really change. It's just sort of static. Okay. And you can't do that much with a static membrane potential. Maybe you can use it to make ATP or something in the mitochondria. That's great. But the neurons have found a way to change the sets of ion channels that are open or closed on the cell membrane in a very dynamic way that will allow that membrane potential to fluctuate very rapidly, and thus allow processing of information and transmission of that information long distances. And so we want to understand how that's going to be influenced by the ion concentrations. So how much sodium, or how much potassium, how much chloride is inside the cell versus outside of the cell. And how the ion channels and which ion channels are open are going to determine how that membrane potential changes and where it stays. That membrane potential can also switch. Sometimes it changes smoothly, okay? Like little fluctuations up and down. And then sometimes it switches into a mode where you have a very sharp change. Okay, in less than a millisecond, the electrical potential shoots from negative 70 millivolts up to plus 30 millivolts, and then back down. Okay, that is a pulse of electrical signal called an action potential, and that is the individual single unit of information processing and transmission in the brain. Okay, so we want to understand how these ion channels are going to allow both this graded changes, slow changes, small fluctuations versus these all or none binary events called spikes or action potentials. Okay. And all of that is going to relate to how sodium channels and potassium channels on the membrane open and shut, okay, and how they might open and shut. Some ion channels are always open, okay, they just are constantly leaking out ions. Some ion channels, if another molecule binds onto them, can open a gate. Those would be ligand-gated ion channels, okay, so you could selectively control when an ion channel is open or closed, okay. Another type of ion channel, you could have mechanically sensitive ion channels and touch. Like in your fingertips, you have one, you put some pressure on it, it opens them up, you get an electrical charge flow. In some cases, you can make ion channels that are actually opened or shut, not by something binding onto them, but the actual electrical potential of the membrane itself, which provides the positive feedback loop, which will allow these all or none spikes to occur. And then finally, we want to understand how the timing of the opening and closing and the kinetics of those ion channels and the sodium and potassium channels are going to be what allows action potentials to take place, how action potentials can transform sort of the amount of electrical excitation in a cell into a pattern of spikes of increasing or decreasing numbers per second. Okay, so this is overall the key concepts. It's a lot to cover, but I think we're going to try to do a good job on it. And first, let's take a look now, though, at the brain itself. Okay, so what's the first thing you see here? Okay, there's a human brain from the Wisconsin collection. Okay, so we've got the main thing that you see here is the cerebral cortex. Okay, so this is a vast structure. In humans, the combination of gray matter, which is where all the cell bodies live, and the white matter, which is just the wires between the cell bodies, takes up about 80% of the brain, okay? And this is going to be organized into really deeply interconnected regions, some of which are going to be specializing for sensory information, so sense of touch, the sense of hearing, sense of smell, sense of balance, okay? And then motor information. By motor information, I mean the planning and execution of movements, okay? And then those are going to be interwoven with areas of where we're associating sensory and motor information together, where we're trying to evaluate a variety of different incoming sources and make decisions and sort of translate, information coming in, our own, build a model of the world, and then take actions or plan actions. So that's a lot of our cognitive abilities, right? So things like language or reasoning would be part of those sort of interconnected areas. And cerebral cortex is not found in all animals, okay? It's really a hallmark of mammals in particular, right? So here's basically a 2-scale representation here of a bunch of different mammals here from human to mouse. What we can see here is there's a bunch of different patterns here. And we've got a lot more wrinkles in the human than a lot of these other creatures, right? Those wrinkles allow an expansion of the overall surface of the cortex of the brain. Because if we think about it, like the cortex of a brain is like a big sheet, okay? And the cortex, the expansion of the cortex over evolutionary time is what has allowed a greater level of cognitive ability in humans than these other creatures like the poor mouse with its smooth brain or the poor rat with its smooth brain. Okay, so in order to sort of increase the amount of surface area that we have in a cortical sheet, what we can do is crumple up that piece of paper. We can crumple up the cortex and we get much more surface area packed into a smaller space. Okay. Now, and we'll talk more about the sort of the gross anatomy in the systems neuroscience portion next week. But let's sort of now zoom in here onto what is the overall purpose of the brain. Okay. It's really just to create adaptive behaviors. It's to create actions that are informed by something in your environment and by things that you have learned about how the world works, right? You can take information in, inform the prediction of how the world works, and then sort of try to make the best action possible in that context, right? So that's going to require taking in information called sensory information from your environment. We've got sensors like our ears or fingertips or our olfactory epithelium in our nose for smelling. We're going to sort of pool all of that information, bring it into the brain, into these sensory systems. We're going to think about it, we're going to transform that information some way through the patterns of the circuits of the neurons within there. And then eventually, we're going to produce a motor output. So we'll have motor neurons that are then going to come out and innervate or like make electrical connections onto the muscles of our body, skeletal muscles, and then we can move in a coordinated way. Okay, so we can really divide the brain, or I guess the nervous system, I should say, into two classes. We've got the peripheral nervous system and we've got the central nervous system. So the peripheral nervous system is anything that's going to interface with the environment or interface with our muscles. Okay, so sensory input, you have to somehow sense a change in the environment, right? You have to sense a vibration of sound in your eardrums somehow. That has to get transformed into an electrical signal. So those primary neurons that interface with the environment, the primary sensory neurons, are going to be part of the peripheral nervous system. They're going to pass that electrical signal into the central nervous system, where we've got a bunch of different brain cells that are going to process it in different ways. And then the final output, when we finally decide what we're going to do about it, that's going to be conveyed by motor neurons that move from spinal column into the muscles, make synapses on the muscles, and cause electrical changes in the muscles that allow contraction of the muscle and repositioning of the body. Okay, so in the human, okay, what part is considered the central nervous system versus the peripheral nervous system? Okay, so we've sort of colored that in here in yellow or in blue. Okay, so we've got the brain is the central nervous system. That's a big part. Cerebral cortex is a lot of it. And we also have the cerebellum, subcortical structures, as well as the spinal cord, okay, that comes down here. That's still considered part of the central nervous system. Now, any nerves, which are just bundles of neurons that come off the spinal cord or come out of the brain by the cranial nerves, those are things that they're going to be directly interfacing with the environment or with the muscles. So that's part of the peripheral nervous system. Okay, so these sort of nerves that extend into the body. There's also a lot of neurons in the stomach and the gut area. You can think of that also as part of the peripheral nervous system, even though it's not really showing up here. So neurons are going to be the cells that are going to be transforming or transferring that information. Okay, and the direction and path that that information sort of transmission is going to take is really going to depend on the shape of the neuron. Okay, and depending on basically the complexity of an organism. So here's a C. elegans. It's just a little tiny 0.5 millimeter long worm that a lot of neuroscientists, it's a common like model in neuroscience because it can do a few little simple behaviors. It only has 302 neurons in it. Okay, and they know exactly where each neuron is. Okay, you can characterize that from every animal has the same number of neurons. Okay, fruit flies maybe got 30,000 neurons in it. It's a lot more complicated. It can fly around. It can defeat you. Or you can usually get away from you, right? If you're trying to hit the smack it on, to kill it. And it has a very developed sort of visual system, good motor control, can react very quickly. Okay. And then finally, you have a human that's got more like, 85 billion neurons in it. And we have a lot more complicated functions there. Okay. So just if we have a large structure like this is called the brain. Okay. And then if we zoom into a brain, there can be smaller structures that are collections of neurons that together accomplish one particular function. Usually this is on the order of like thousands of neurons collected together in one little spot. That's called the ganglia. Okay, so fruit fly have ganglia. Humans have got ganglia too. Got the dorsal root ganglia, which are going to be involved in sensory peripheral nervous system processing. We also have things like the basal ganglia within the brain itself, which are really involved in motor coordination, motor control. Damage to that can really screw up a person's ability to choose actions and make coordinated motor movements. But then some creatures really, they don't really have much at all. They just got 300 nerves together, just a collection of simple nerves like a jellyfish or a worm. So they might have, this is sort of considered the brain here or a ganglia here. And then along here is just a bunch of wires of their peripheral nervous system. So anytime we're in the brain or we're going down a neuron, okay, or through nerves, we're going to have long distance. We have to send information long distances. And in order to do that, we have to use electrical signals. Okay, it's the only way that we can send something very quickly. I'm talking, in tens of milliseconds, you can send a message from your brain down to your foot. Okay, that's not going to happen by a circulation of a chemical through your blood, right? But over very short distances within the nervous system, we do rely on chemical communication. And this will be the release of molecules called neurotransmitters across tiny little gaps called synapses between two neighboring neurons. Any questions? Yes. Yes, the release of chemical signals is controlled by electrical signals in the neurons, and we'll go into that. Okay, so there's three major classes of neurons, okay? And they can be divided into the peripheral nervous system on the left, these two, and the central nervous system on the right, okay? Now, let's look at the peripheral nervous system first. We can divide that into the sensory neurons. These are the ones that are taking in information from the environment, and the motor neurons. These are ones that are going to take our plans for action and turn them into actual contraction of our muscles and movement of the body. And then on the right-hand side, we've got interneurons, and this is basically any neuron in the central nervous system you could call an interneuron because it is not directly interfacing with the environment. But by the way, I just want to be clear, interneurons still can carry information that is relevant to sensory signals and relevant to motor signals, right? If I'm planning a motor action, but I'm not actually contracting the muscles, I'm an interneuron, but I could be a motor-related interneuron. If I'm sort of, trying to generate the perception of a smell or the feeling of an object, that's going to be conveyed by electrical signals within interneurons in the brain, but those could be sensory interneurons. Okay. But within the peripheral nervous system, we can sort of divide the cell up into these different parts. Okay, so all the cells, all neurons are going to have a cell body, right? This is where the nucleus is. This is where our genes are stored, right? This is where proteins are going to be generated and then transported down the processes of these neurons into various specialized endings. Okay, so we always have sort of an input end and an output end of a neuron. Where is the information coming in from, and where is it coming out of? Okay. In the sensory neurons, the information is going to come from specialized receptors. Okay, these are just endings of the neuron that have been sensitized to some aspect of the environment. It could be a chemical. So if you're a smell or a taste, right, that is caused by chemicals of particular shapes binding onto receptors of particular shapes, these ligand-gated receptors that will then open channels that will cause an electrical conductance to take place in the nerve ends. Okay, or in this case, in this sort of example here, these are receptors in the skin. These are some sort of pressure-sensitive receptors. If you bend the skin, it causes some shear force. It's going to cause the opening of mechanically gated channels and cause an electrical signal to initiate here by the movement of ions across the plasma membrane of the cell. Okay, so now if we look here, we also have this sort of path. The dendrite is always what's taking in the information, that's taking in signals from either the environment or from other neurons. Okay, so this end's called the dendrite. Okay, and then there's going to be an output end. This is where electrical signals are going to get sent out of, and this is called the axon. Okay, so electrical signals are going to travel from the dendrite to the cell body and then down the axon to synaptic endings. So these are places where the electrical signal goes to and it gets turned into the release of specific chemicals like neurotransmitters. This could be glutamate, it could be dopamine, it could be GABA, it could be a whole range of different sort of chemicals that can be released. And that's going to then sort of transmit the message to the next set of neurons. In the motor systems, we have sort of the same features here, but they're structured differently, right? So now we've got a bunch of like fingers that are growing out here, dendrites, almost like a tree branches that are sort of trying to absorb light. Actually, in this case, they're just trying to absorb chemicals falling onto them from other neurons, synaptic endings. Okay, so these are taking in the information in the dendrites. They're sort of, that information is getting added up in the cell body, and then the electrical signal is going to travel down the axon to these muscles, okay, where it will release at the neuromuscular junction. It will release a chemical called acetylcholine, it will open up acetylcholine receptors and then cause a depolarization of the muscle, cause contraction of the muscle. Okay. You see this weird stuff wrapping around it? These are wrappers that are insulating wrappers that are made by Schwann cells, okay? And the Schwann cells basically are going to allow a faster propagation of electrical signals down this membrane. We'll explore exactly how that does that later. And what we can see here is, so these myelinates, so this is called this wrapper, it's almost like wrapping the membrane in a bunch of saran wrap. This wrapper insulates the membrane, okay? And in between, it insulates it in little steps here, okay? You have little gaps between each wrapper, and these are called nodes of Ranvier, and electrical signals can actually skip over from node to node to node through a process called saltatory conduction, and that's going to really accelerate the pace at which electrical signals can propagate. Now, the sensory neurons are a little bit weird, okay? They can actually cause an action potential or a little spike of electrical, a wave of electrical potential that can start over here at the dendrites and then propagate all the way through here to the cell body and go past the cell body down the axon to the synaptic endings. In the case of the motor neuron, the action potential does not start in the dendrites. It starts right here at the beginning of the axon, okay, called the axon hillock. And this is typically how it works. This is how it works in motor neurons, and this is how it works in all the neurons in the central nervous system. All the interneurons are going to be integrating information from their dendrites and then initiating the action potential right around here, right at the beginning of the axon. And that will then propagate down this axon onto here. So more generically, within the central nervous system, in the interneurons of the central nervous system, we still have a cell body. We have dendrites that look just like the motor neuron dendrites. And then these axons are going to go out, and they are not going to travel to muscles. They're going to travel to other neurons within the body or within the brain. Those connections might be myelinated. They might be wrapped up. They might not be wrapped up. It really just depends on where those axons are going and how far they're going. But in all cases here, information is coming into the dendrites, getting integrated in the cell body, and then an action potential will be generated here in the axon that will go down to the synaptic endings. So this is just some sketches. The sensory neuron and the motor neuron look about like this, okay? The interneurons actually, though, have insane amount of diversity within their structure within the central nervous system because they have so many different information processing functions that need to be accomplished. Okay, and here's just, you know, a very basic sketch of a few sort of shapes of these neurons, right? And here is the shapes or the morphology of the neuron. So what they're shaped like is really going to give you really good clues into the specific functions that they accomplish. Now, this is sort of the classic example of what a real neuron looks like in the brain. Okay? This is filled by a silver stain called the Golgi method that was developed in 1880 by Camillo Golgi. And this was basically stained, and you've seen one neuron here. Okay, what do we see in this neuron? We see a cell body here, and we see all these arms coming off in different directions. Okay, on those arms, so those arms, oops, what are these arms? These arms are the dendrites. This is where information is going to be coming in. Okay, on the tips of those arms, all I guess studded along the length of those dendrites, what do we see? If we zoom in to this spot or this spot, the A or B spot, we see these little protrusions. These are called spines. These are little compartments where chemical signals are going to fall onto those spines, and you will get an electrical and chemical signal just restricted within that individual spine. Those are the synapses. This is where the synaptic input comes. But where are the axons? Where are the actual inputs that are onto those? You can't see them in this because we haven't stained all the neurons. We've only stained one neuron here. Only one neuron is black, okay? Everything else is transparent. if you were to stain all the neurons, it would just look like a complete black mess of like squid ink pasta. Okay, you'd be completely unable to see what's going on. So you just have to imagine there's a bunch of invisible axons traveling over here and making little connections on each one of these little spines has its own axon, another axon that's connecting to it, and is going to release chemicals onto that spine. Okay, so that is where all that chemical input is occurring. Now, what about the axon of this cell? Where is that? Usually they're kind of hard to see. I think probably there's one axon, this tiny, thin little thing coming off down here that then disappears. It's probably the axon. And that's where the electrical signal is going to be sending out of, and it's going to be going a long way away. Okay. A lot of times when you look at sort of a drawing of a neuron, they don't really show you where the axon goes because the axon could go so far that they had to like make the map of the neuron. They had to take a slice of a brain. The axon is usually traveling way outside the slice. It goes going a long distance. So this is one example. This is from the cortex. This is called a pyramidal neuron in the cortex. So the cerebral cortex, it's called a pyramidal neuron because the cell body looks like a little pyramid. Okay, so it's got lots of synaptic inputs. It has an electrical output that goes down the axon, and it's going to make synapses onto thousands of other neurons. Okay, and there's billions of these neurons in the brain, right? So it's actually quite a complicated mess in order to figure out how it all works. There's some biology of neurons. Usually, you know, so here's in black are the dendrites here. This one is a particularly crazy dendrite, right? And then in red here is the axons that are going somewhere far, far away. So the morphology of the study of the morphology of neurons and how it relates to their function was really developed by this guy, Santiago Ramon y Cajal, Spanish scientist at the turn of the last century, who used those Golgi stains and made these really beautiful drawings of all these different parts of the brain. And, you know, basically sketched out these little spines, right? And he hypothesized that neurons' structure was intimately related to their function, and that information would flow in a direction from the dendrites into axons and then be sent to other parts of the brain that would then process that information based on the shapes of their dendrites and axons. So he really, you know, developed this theory that the brain is an information processing. So they didn't have what a computer was, but it was some way of processing that information, and that how those neurons were wired up would define how that information got processed. Okay, so let's sort of take a sketch of like this. So how can neurons actually get wired up into circuits? And what are the definitions of that? So we've got our neuron that we just saw here, right? We've got our cell body or the soma. Okay, most of the organelles are going to be in the cell body or the soma. There will be some organelles, mitochondria sometimes will often appear in these areas here, these tips, these presynaptic terminals, because you need to have a lot of energy out there as well. Most of our neurons are going to have dendrites where the information is coming in from. And the axon is going to be a long, thin wire that's going to go out to diverse locations and make synapses, usually on many, many other neurons. Okay, not just one here, like it's described. But imagine this branching and going to many different neurons. Okay, so right here at the base of the axon, right where it starts, there's going to be a swelling called the axon hillock, a tiny hill. And that's named because it's just a little bit it looks like a little hill coming off the cell body. Okay. And that's going to be this place where our action potentials are initiated. The electrical signal will fire down here. And then it's going to come to what's known as the presynaptic terminal. Okay. There, the electrical signal is going to cause the release of vesicles, little sort of balls full of neurotransmitters, of chemicals that will then get released onto the next cell. On the next cell, there will be receptors that are sensitive to that neurotransmitter, which will then cause a change in the membrane potential on this postsynaptic side and potentially lead to an action potential in the downstream cell. Usually, you're not going to be able to get an action potential from the action of just one of these synapses, though. You're going to have to add up the combined functions of hundreds or thousands of synapses at the same time in order to generate an action potential. So that's sort of the sketch of what it looks like. What about like an actual brain? Oh, it's so dark. Maybe I can turn that down just briefly. So here is a fill. So this is a technology that was developed a few years ago. This is called basically a cleared brain. So instead of like taking a slice of the brain and trying to figure out where the axons go, you could take an entire brain and plasticize it and then remove the fats and then create index matching. And you can actually make the brain totally transparent when it's out in sort of a dish. And then you can trace the overall shape of individual neurons and where their axons go within the brain. How is this done? They used a virus, they injected a virus into here that contains a green fluorescent protein gene that makes the neurons produce a fluorescent protein from jellyfish. Now the neurons are glowing green. And we can see here, one of these cells up here, these are the pyramidal neurons of the cortex that we saw earlier. Remember that cell fill that had all the spines on it? This guy. This is the shape of one of those neurons. And now we can go over here. And one of these guys is that neuron. So that entire elaborate sort of dendritic structure that you saw before is all local. It's all contained right up here. Whereas the axons are actually traveling very long distances to other parts of the brain, to the pons here in the midbrain, and also to the cerebellum and other sort of areas all over. So these axons are going to travel long distances. Dendrites are typically going to stay local, okay, because they are going to work by, their inputs are going to be sort of integrated as graded potentials, changes in membrane potential that don't trigger an action potential on their own. And the long distances are going to be, information is going to be conveyed by the axons, which are going to be the pathways in which action potentials can travel, which is a different sort of mode, this all or nothing mode. Okay, so what happens, right, when we travel down the axon, we're going to make a synapse onto another neuron. A synapse is just a junction between an axon and a target cell. Okay, we've heard of synapses before, right? The immunological synapse that cytotoxic T cells are going to make on other cells where they get in close apposition to one another and dump granzymes and stuff on it to kill the cell. Okay. In this case, synapses are going to be found between axons and other cell bodies, or sometimes on the spines and sometimes just along the dendrite itself. So we're going to pass information from the presynaptic terminal. That's the side that's going to be releasing the chemical or neurotransmitter. And they're going to land on a postsynaptic terminal, okay, which is the side that's going to have the sensitive receptors for those chemicals. So that's the standard method of transferring information between two neurons, is this chemical neurotransmission at synapses, synaptic transmission. I have to say briefly, there is you can also have direct electrical passages, of course, through gap junctions. This happens in some areas of the brain, like in the peripheral nervous system, like the retina. But the primary mechanism is this chemical neurotransmission. And how many synapses do we have? It really depends on the cell and the morphology. You can have up to 250,000 synaptic inputs in the Purkinje cells of the cerebellum. Okay? Or in other part of the cerebellum, the cerebellar granule cells only get 3 to 5 synaptic inputs total. So very different size and morphology. If you want to say what the average number of synapses onto a neuron is in the brain, say like one of those pyramidal neurons that we looked at, let's say on average about 10,000 synapses per neuron. Okay. And so if we've got 85 billion neurons, we've got about a quadrillion number of synapses in our brain, really quite a few to count. This is what an electron micrograph of a single synapse looks like. We have a presynaptic terminal here. How can we tell it's a presynaptic terminal? So this is a slice, a thin slice of a one neuron talking to another neuron here. We have these little holes here, these little circles. Those are the vesicles that are full of neurotransmitter. We've got these other structures here with all these little wiggles in them. Those are the mitochondria, which is where we're making the ATP. And those neurotransmitters are going to, those vesicles will fuse with this membrane, dump out their neurotransmitter here, and they're going to land on receptors on this other side. So this is the receiving cell. This is the releasing cell. And those are going to open up different channels and cause a bunch of second messengers to sort of get activated here. And you can sort of see this density, this dark sort of stain here on that receiving end. That's all of the different scaffolding proteins that are holding those channels in place and that are going to allow signaling to take place in the postsynaptic cell. So these spines, let's talk a little bit about spines. This is where we're going to be receiving neurotransmitters. Okay, so this is along the dendrite. You see these little protrusions. They have different shapes. You could call them mushrooms, thin spines, short, stubby spines. This is the location and the changing of spines. It's thought to be where memory is encoded, right? So if I want to learn something new, okay, and I want to keep it in memory for a long time, okay, if I want to remember it the next day, okay, I'm going to have to change some pattern of these spines and the shapes of these spines, the strength of those spines. I might have to delete some. I might have to add some new ones so that I can store that memory. Okay, so that's the physical substrate of memory formation. And these spines are constantly changing. Some are just going to be new ones growing, ones that are going away. But then there's also some that are very persistent for months at a time. And those are the ones that are really encoding our long-term memory. Spines also, so spines, at the tips of these spines is where the neurotransmitter is going to be released onto, and you're going to get an electrical signal here that will, and some chemical signals in the postsynaptic side sometimes too, maybe calcium flowing in, and that will spread a little bit, okay, into the shaft. Depending on how sort of skinny the spine is or how sort of thick the opening is, how close it is, you can get more or less spreading of that electrical and chemical signal into the dendrite itself. Now, if you didn't get any spreading of electrical or chemical signals into the dendrite, you'd never be able to add up all of the activity coming in from all these spines in order to generate an action potential. So you have to have some, but the spines have like basically a partial compartmentalization of signaling and allow individual spines to be strengthened or weakened without affecting the neighboring spines. Okay, any questions on this stuff? Yes.
Okay, that's for some folks, you can remember it for the rest of your life.
Mhm.
They don't remember that. How does that impact the attention of spine?
So that's a great question. So the more, oftentimes the more traumatic event that occurs, the more salient, we might call the more salient stimulus, something that is deemed most important. Okay, that can the salience of a stimulus can be sort of signaled by certain neurotransmitter systems like dopamine or norepinephrine that are going to basically modulate the plasticity of these spines, so how well they can change. So you may have a short window of time after that event takes place where the spines are super malleable, they're super plastic, they can change dramatically. You can get a major reorganization of them just for a narrow window of time. Okay. Secondly, I think in terms of like why sometimes it's suppressed versus sometimes it's very I guess it's persistent, that traumatic memory is persistent. I think it depends on the reinforcement of that memory over time. So as, and we'll talk about this maybe in the systems lecture, as neurons that basically are co-active together are going to tend to get strengthened together. The connections between neurons that are co-active at the exact same time will tend to be strengthened. Neurons that are active, the connections between the neurons that are sort of not active together, they're active, but they're not in sync with one another, those will tend to get weakened. So by sort of replaying an event in your mind over and over, you're re-triggering the sort of set of neurons that are representing that memory, okay? And you reinforce the strengthening of the connections between those by replaying the event over and over and over again. So that would cause a strengthening. There's probably other mechanisms that are related to not wanting to feel the trauma, other brain systems that are probably suppressing that memory and just trying to prevent those patterns from being played. And the lack of replay of those patterns could probably suppress that memory by reducing the strength of the connections that represent that memory. Any other questions? Okay. So I'm going to briefly talk about glia cells. Okay. So we've got some neurons here. Okay, here we got a bunch of Purkinje cells here. They've got these are really elaborate dendritic arbors. And then we've got these other cells here stained in red that are glia, okay, or red and blue. Okay, the glia are supporting cells, and they have a variety of different functions. Okay, we've got ependymal cells. Okay, so we can sort of here's a gradient here from the central nervous system to the peripheral nervous system on the left side to the right side. So sort of the core of the central nervous system, maybe near the blood. There's going to be a barrier with the blood, or within the ventricles which are like these, like almost like little oceans of fluid inside the brain. Okay, there we're going to have ependymal cells, which are going to form an interface between the ventricles and the rest of the brain. Okay, so those are glia and those are going to help with signaling things like, measuring the overall osmolarity of your blood, or the pH of your blood, because they're going to measure that through the pH or the osmolarity of the cerebrospinal fluid, which is what's filling the ventricles. Then we've got astrocytes. These have a variety of different functions. They are going to provide physical structural support for the neurons, right? Neurons have got like these really elaborate arms everywhere, right? And pretty skinny, right? So you want to have some sort of scaffolding structure that holds them together. Okay, so astrocytes are going to do that. They're also going to help form the blood-brain barrier. So blood does not get into the brain. That's very good in a way, because there's a tight seal. Okay, that's to help to prevent like bacteria or any possible pathogens from getting inside the brain. If you have a tight barrier, that's going to prevent that from happening. It also makes it difficult to treat when things do get into the brain, like meningitis or something. The pathogens get into the brain. It can make it very challenging for the immune system to fight it off because of the blood-brain barrier. So anything that is going to get into the brain needs to pass through specialized transporters on the blood-brain barrier. So not everything that you eat or every drug that you take can get into the brain. It has to be modified into a form that can pass through the blood-brain barrier if it's going to have an action on your neurons. Yeah.
About like blood barrier, like what's it called, the cerebrospinal fluid? Is that part of the brain barrier, or?
So the cerebrospinal fluid is so that there's still a barrier between I'd say the ependymal cells make the barrier there, and the cerebrospinal fluid is going to be able to pass material into and out of the blood, but the actual cells are not going to be able to get in and out. But certainly, like the small molecules and pH stuff can get in and out. Okay. And then astrocytes also are going to secrete things like neurotrophic factors. These are things that are going to help neurons grow, nurture them. They're kind of like basically supporting cells, structural and chemical support cells. They're also going to suck up leftover neurotransmitters. If your neurotransmitter gets released, okay, you're going to want to recycle that neurotransmitter. So astrocytes stick their tiny little fingers, okay, right along the sides of synapses and have transporters that are going to basically vacuum up stuff like acetylcholine or glutamate from that spills out of that synapse, and they will repack that and send it back into the neurons so that it can be re-released. Then we've got microglia. These are these tiny little cells that sort of act like the scavengers that are going to pick up like decaying stuff. And the microglia are actually by far the most numerous cells in the brain. We don't know that much about them, but if you poke like you have trauma in the brain, maybe from a concussion, right? You'll see, or by poking something into the brain, like, I don't know, an electrical wire, that if you're trying to build a brain-computer interface, one of the big problems is if you stick electrical wires into the brain, at the beginning, you'll be able to hear electric signals really well, but after a month or two, all the microglia come around and start coating those wires to seal them off and recreate a protective barrier, right? So that means you no longer have electrical access, and that's a problem with, maintaining brain-computer interfaces for long periods of time. So microglia are sort of the scavengers here, and sort of try to act as a protection on the brain. And then we've got these Schwann cells here and oligodendrocytes. These are two cells that are basically the same kind of cell, just in two different locations. So the oligodendrocytes are what are going to myelinate or wrap around the insulation on axons in the central nervous system, whereas the Schwann cells do the exact same thing in the peripheral nervous system. Okay, and we can sort of see this here. Okay, here is a electron micrograph of an axon. So this is a slice through an axon, right? So the axon is very long and thin. We're taking a slice like a salami slice. And then you see all of these sort of wrappings around it. This is the myelin of a Schwann cell, the nucleus of which is here, that has sort of ensheathed it many, many times. Okay. Right. In here we can see how the microglia here are really outnumbering our pyramidal neuron and the astrocytes, which are labeled here in green. And then over here we can see how ependymal cells can form a barrier between the blood or the cerebral spinal fluid and the brain. Okay. Another thing that ependymal cells do is they are going to actually be the source of neural stem cells. So most of your cells, most parts of your brain don't create new cells over time. There's a few areas, a few niches, which would continue to create neurons over the course of your life. But you actually are born with like twice the number of neurons that you have by age 2, you have half the number of neurons that you're born with. Okay, that's a programmed strategy to help wire the brain up. And those are basically all you've got for the rest of your life, except for a few small areas of the brain where you continue to have some growth in brain cells, and that is going to be accomplished by these ependymal cells. Okay, any questions on glia? Yes. Yes, absolutely. So the microglia in particular are going to proliferate and move towards areas of inflammation and damage. Yeah. Yeah, yes. So astrocytes are going to be protectors, are going to be the scaffolding. They also are going to be releasing chemicals that allow that growth to take place during development, and sort of provide factors that are going to basically tell the cell to stay alive, keep working. In the absence of some of those factors, these neurotrophic factors, the cell can end up killing itself and undergoing apoptosis and giving itself up to its neighbors, which is sort of the process, which is really a process in development. So astrocytes play a very large role in the wiring up of circuits as the brain grows throughout development, but also in the maintenance of those circuits as an adult as well. Yeah.
So like astrocytes are only found in the central nervous system, right?
Yeah.
So how will like the neurons up like the peripheral nervous system get like structural support and stuff?
Okay, that's an interesting question. Okay, so the neurons, so they're usually in bundles of usually you don't have a single neuron axon that's going out by itself. Usually they're bundled in a nerve. Okay, and those nerve bundles are going to be, I'd say, really structurally supported by this myelin sheath as well, and just by being attached to all the other nerves next to them. Yeah. Okay. So now let's talk about this great mystery of neurons, which is they are these they're bioelectrical devices, right? They're made of like fat and salt water and proteins, right? But somehow they can transmit electricity in very precise ways. Okay. How does that electrical and use that transmission of electricity and that integration of signals in order to make computational processing of information and control, our body. So how does that take place? Really, there's going to be a couple different topics here. One is the resting membrane potential. One is the action potential. This is a sudden rapid change in membrane potential. And that is generally going to go in one particular direction and it's the action potential, but not normal membrane, but not small fluctuations in membrane potential can propagate very long distances. So how does this take place? How is this even possible? Okay, it's going to come down to the membrane here. So imagine, we have a plasma membrane. Every cell has a plasma membrane, right? A plasma membrane is made up of fats. It does not allow any ions to pass through it, okay? Unless there is a specific channel that is open for that ion. An ion is simply an element, right? That is charged, okay? So it's either missing an electron or two, or it's got an extra electron or two. Okay, so it can be positively charged. So the positively charged ions, these cations, okay, are sodium, potassium, calcium. Those are our big three for that, okay? Sodium and potassium are the ones that are going to be most important for the membrane potential, whereas calcium is going to be the most important for actually triggering second messenger signals inside the cell. Okay, so those potassium ions and sodium ions have a positive charge of 1. Calcium ions have a positive charge of 2. So monovalent cation, sodium and potassium. Divalent cation is calcium. Okay, we also have some negatively charged ions, the most important of which is chloride. Chloride is negatively charged, right? So, table salt is just sodium chloride stuck together, okay? When it's dissolved into a solution, the sodium and chloride are going to dissociate from one another, okay? They're going to want to be close to one another as long as they are in the equal balance, but sometimes there'll be some imbalance of the amount of sodium and chloride in one area or the other. And that is going to be accomplished by the movement of specific ionic species across this plasma membrane. Okay, so in most cells, okay, there is going to be a negative charge inside the cell relative to the outside of the cell. Now, this is accomplished by a number of factors. One factor is proteins inside the cell tend to be more negatively charged than proteins outside the cell. Proteins outside the cell are usually like neutrally charged, sometimes positively charged. Proteins inside the cell are negatively charged. Why is that? It's really based on what amino acids are part of the protein and at what pH they are positively or negatively charged. Okay, so we're just gonna these proteins, okay, these anions that are they're stuck inside the cell. They can't get across the cell membrane. They're too big for an ion channel to let them flow across. Okay, so first of all, we have this sort of reserve pool of these negatively charged anions inside the cell. It's important to have them there because we need to have a negatively charged inside of the cell so that we can make ATP in our mitochondria. There's some that uses that gradient to do that. But so they're sitting here. Okay, so this is going to cause the inside of the cell to be a little bit more negative than the outside of the cell. And what would that tend to do? Do opposites attract, or do they opposites attract? Positively and negatively charged ions want to come together, right? So if you have the inside of a cell that's got some negatively charged anions, that's going to produce an electrical gradient between the inside and the outside of the cell that want to pull some positively charged ions into the cell. Okay. However, the electrical gradient is not the only thing that matters for motion of a ion across a membrane. Also, that relative concentration of a particular ion inside or outside of the cell is also going to have a huge impact on what direction an ion might want to pass through a membrane. Okay, so the overall membrane potential here is the difference in electrical charge along the surface of the inside part of the membrane versus the surface of the outside part of the membrane. Okay. And what we call the resting membrane potential is what that's normally set to. If not much is happening, if we're not changing anything, just the default state of a cell membrane is usually around -70 millivolts, and that is the resting membrane potential. So that means the inside is -70 millivolts more negative, 70 millivolts more negative than the outside of the cell. Okay, a volt is just a measure of electrical charge difference. So there's this separation here of positive and negative charges across this very thin membrane. Okay, how do we actually determine that membrane potential? It's really going to be dependent on two things. What is the concentration of ions inside and outside the cell? Okay. And secondly, how permeable is the membrane to those ions? Okay. So each ion, okay, can only pass through the cell if there is an ion channel that allows that cell to pass and that ion channel is open. Okay. Now, there's a great variety of different ion channels in the body in these plasma membranes. You can have ion channels that are selective only for potassium ions. Okay, only potassium can pass. You can have ones that are selective only for sodium ions. Only sodium can pass. You can have ones that are selective for cations. Okay, so sodium and potassium can pass. You can have ones that are selective for divalent and monovalent cations that allow sodium, potassium, and calcium to pass. You have ions that are chloride channels that only allow negatively charged chloride ions to pass. Okay, so there's this great diversity of potential ion channels. All of these are encoded by our genes. Okay, but most of those ion channels are not expressed on the plasma membrane. Okay, the majority of the plasma membrane at rest, okay, ion channels that are just open all the time, most of the channels there are going to be potassium channels. We're going to have some sodium channels, mostly potassium channels. Okay, and that will make it so that the membrane is more permeable to potassium than to sodium. Okay, so what do I mean by permeable? How easy is it for a molecule to move from an ion to move from one side of the membrane to the other side of the membrane? You've got to have holes. The more holes you have for potassium, the easier it is for potassium to get out, right? If we remove all the potassium channels, then we'll have no potassium permeability. Okay, so now if we see here in this graph, we have one other thing here. We've got that sodium-potassium pump. The sodium-potassium pump is always running. We saw it in the kidney, right? We see it in these cells as well. It's always pumping in 3 sodiums in and 2 potassiums. Sorry, 3 sodiums out and 2 potassiums in. What will that tend to do over time? So we're spending a lot of ATP constantly pumping sodium out and potassium in. Why do we do that? It's so that we have an imbalance in the concentration of sodium and potassium inside and outside of the cell. So the sodium outside the cell is about 150 millimolar. Inside the cell, it's 15 millimolar. That's a 10-to-1 ratio, right? Potassium is 5 millimolar outside the cell, 140 millimolar inside the cell. That is almost like a 15-to-1 ratio, or 30-to-1. I don't know. It's an even bigger number. 30-to-1. Yeah. Okay, and then we also have an imbalance here of chloride ions, 120 millimolar outside versus inside. And we've got some anions, negatively charged anions sort of sticking in here that can't pass between the membrane. Okay, so we can see this in this plot as well. How does this is sort of a chart here that's going to show you the distribution of different ionic species in the blood, in the interstitial fluid, and in the intracellular fluid. So one of the first things that you can see from this chart is that the blood and the interstitial fluid are usually almost identical, right? So sodium, huge in the blood, huge in the interstitial fluid. Potassium, low in the blood, low in the interstitial fluid. Okay. The big difference is going to be between, and the important difference for the membrane potential is what is the concentration between the interstitial fluid and the intercellular fluid. Okay. So we just saw sodium and potassium. That is a huge difference between those two. What about calcium? This is also another very important molecule. We see, a small amount on the outside of the cell, and you can't even see the inside of the cell. Calcium actually has the greatest difference in terms of ratio of concentration between the inside and outside of the cell. The outside of the cell is a few millimolar, 3 millimolar, 2 millimolar. The inside of the cell is like 50 nanomolar. It's really 100,000-fold difference in the concentration inside the cell and outside of the cell, which is going to make any calcium flowing into the cell cause a huge change in the concentration inside the cell. That's what's going to make calcium such an effective second messenger molecule. Okay, and then the other one that's sort of relevant that can influence membrane potential is going to be chloride ions here. Okay, a lot outside, not many inside. So let's sort of look here and see how the membrane potential is established. Okay. So we have this asymmetric distribution of sodium on the outside, more sodium on the outside, more potassium on the inside. And we have most of the, and that's being maintained by the sodium potassium pump. Okay. Now, what we need to do is we need to have potassium channels in the cell membrane. And what will that then do to the membrane potential? Okay. So if we have a bunch of potassium selective channels inside this membrane, we have a lot of potassium on the inside and not much potassium on the outside. What will want to happen? There's a chemical gradient here. Potassium wants to leave the cell down its chemical gradient because it wants to equilibrate the relative concentration of a single of that ionic species inside and outside the cell. Okay, so that chemical gradient is pushing potassium ions out of the cell. Okay, but wait, okay, there is the potassium, if the potassium ion was not charged, if it was, neutral, then it would just flow out and it would equilibrate equally on both sides, because the only thing that would be influencing its motion would be its concentration difference. But it's electrically charged. So there is a second feature here. The electrical charge is going to want to pull the potassium back into the cell, right? Because if the inside of the cell is sort of negatively charged here, there's these anions sitting here, it can feel, the potassium ions can feel that, and that's sort of tugging the potassium back into the cell. However, there's such an imbalance in potassium. There's so much potassium inside the cell versus outside of the cell. The net flow, okay, is going to basically be out up until you get a buildup of negative charge inside the cell from the potassium ions leaving, such that the electrical membrane potential that pull from that negative charge balances the force of the chemical gradient pushing the potassium out. So these two things are going to be in balance with one another. And there'll be some particular membrane potential, okay, where the flow of potassium out down its chemical gradient is balanced by the pull of the electrical gradient back in. Where is that? What electrical potential is that at? Okay. It is at negative 90 millivolts. This is called the equilibrium potential for potassium. Okay, so this is the level where the inside of the cell is negative 90 millivolts, and you have all the potassium channels open in the world. You're going to have no net flow of potassium into or out of the cell because the chemical and the electrical forces are balancing one another. This is called the electrochemical gradient. Okay. So if we went sort of more negative than then we would have a net flow of potassium into the cell, because it's more negative, it would be pulling in the potassium more. If we have potassium move into the cell, what would happen? That will start to bring the membrane potential up to negative 90 millivolts. If we were to go above negative 90 millivolts, then the inside is not as negative. It's not pulling on the potassium as hard. The potassium will leak out and tend to bring the electrical potential back down to negative 90 millivolts. If only potassium channels are open, then, and the concentration difference is what it is, okay, which is maintained by that pump, then the membrane potential would be negative 90 millivolts. But we don't only have potassium channels. There also can be sodium channels. Let's imagine what would happen with sodium channels if we only had sodium channels. Okay. We've got a ton of sodium outside the cell. We have not very much sodium inside the cell. The inside of the cell has got a negative charge on it. What's the sodium going to want to do? It's going to want to go in. It's got a chemical gradient that's trying to pull it in and an electrical gradient that's going to be pulling it in. Right? There's a lot of what we'd call a driving force that's driving that sodium into the cell. Okay. So when the sodium goes into the cell, what would happen? There's no chloride channels available. It's going to leave behind our chloride ions. Okay. So it's going to now make the inside of the cell more positive than the outside of the cell. Okay. Don't know why that's in that direction. It's not correct, but anyway, so here we go. So at some level, right, if we have enough sodium coming into the cell, eventually the inside of the cell is going to build up a positive charge because we keep pushing sodium inside the cell up until a point where the electrical potential inside the cell repelling the positive ions out is going to balance the chemical gradient, pulling the sodium in. So that potential is plus 50 millivolts. Okay. So somewhere in between, so the reality is, in a real cell, we have a large number of potassium ion channels open and a small number of sodium channels that are open. And so the actual membrane potential is somewhere in between this equilibrium for sodium ions and potassium ions. And it's usually right about here, around negative 70 millivolts, okay? Because the more permeable the membrane is to a particular ionic species, the more influence that ionic species has on the membrane potential, okay? Yes?
We count the electrical or fluoride ion in amino acid calcium or potassium. So sodium ion.
Right. So I guess to answer that question, when you're talking about the electrical potential, okay, you have to add up sort of the influence of all of the different ionic species together. They all are sort of combined together to create one particular number for what the membrane potential is. How negative is the inside versus the outside? Okay. For any specific ion, okay, every ion is going to be influenced by that specific membrane potential, okay, which is the combined sum. But the chemical gradient, okay, is specific to each individual ion. So potassium doesn't care what the relative ratio of sodium is, inside or outside. It doesn't care what the relative ratio of anything is, except for itself, okay, in terms of its chemical gradient. But it does, for the electrical gradient, you have to look at what is the overall membrane potential, which is all of that stuff added together. Okay. So sort of to review this sort of thing. So we have an uneven distribution of potassium and sodium inside and outside the cell, okay? That's going to establish our resting membrane potential. And then we have more potassium inside, we have more sodium outside. We're going to maintain that concentration pumps all the time, burning ATP. And that concentration gradient is a chemical potential energy that's going to want to move ions if ion channels are open for that specific ionic species. When an ion is going to move through that channel, that is going to change the electrical potential, okay? And it is going to tend to create an imbalance, a change in the membrane potential. Since we have many more potassium ion channels open than sodium channels open, potassium will tend to diffuse out of the cell, down its concentration gradient, until the point at which the inside of the cell is sufficiently negative so that the electrical gradient balances the chemical gradient. Okay. We can look at the, we can also look, we don't have to look inside a cell. So now let's consider the case of just a beaker, okay, that has a plasma membrane in it, and we have only potassium channels within that. Okay. So we can now dissolve some amount of potassium chloride in one side, dissolve a different amount of potassium chloride in another side. Okay. How do will there be a membrane potential between these two sides? If you have no ion channels open, there'll be no difference. If you do have ion channels open, but specifically you have only potassium ion channels open, then potassium ions will flow from the area of high concentration to the area of low concentration, okay? Up until an electrical charge builds up between these two, electrical potential difference builds up between these two sides, such that the electrical charge sort of pulling the potassium back into the inner chamber balances out the chemical gradient. At what point can we, how do we actually come to the number? What is the actual value of that electrical gradient that will reach this equilibrium, this balance between the inside and outside? This is defined by an equation called the Nernst potential, okay? And it's equal to a constant number here, which is 62 millivolts. It's related to like a bunch of different sort of physical constants. And it's also related to the ratio of the inside and outside concentration of the potassium. So we have potassium outside is 5 millimolar. We have potassium inside 140 millimolar. Divide those two by each other. Take the log base 10 of it, multiply by 62, and we're going to come up with negative 90 millivolts. So what's a log again? It's just what number you have to raise 10 to in order to get this ratio. So this is like 1/30th, you have to raise 10 to like the negative 1.5 power to get to 1/30th. It might be, so that's the equilibrium potential for potassium. This should just sort of, I guess, help for studying. And then what about for sodium? Okay, so with sodium, if we have sodium 150 millimolar on the outside, 15 millimolar on the inside of the cell, we have only sodium channels open here. Then we have a ratio of 150 on one side and 15 on the other side, right? So we divide the outside by the inside. 150 divided by 15 is 10. What power do we need to take 10 to get to 10? We take it to the first power. So log of 10 is 1. 1 times 62 millivolts is 62 millivolts. Okay. Okay, that's the Nernst equation. Kind of complicated. Any questions on this? I'm going to stop with Nernst equation because we're going to talk about the action potential next time. We're going to see how this dynamically changes, but this is how you're going to structure, or this is what is defining the resting membrane potential, is the relative ratio of the ionic concentrations and what the permeability of those ion channels are. Okay, thank you.
So that's ideal. So like in reality, like, so in reality,
Hey, how's it going?
some stuff are-
Yeah, I should be around. Stop in. Yeah, HNB 228. Yeah. Well, you're welcome.
Doesn't matter. Or I get not SAC, I think. How can it matter? Like, how can you get negative 90? Probably like, I'm not sure you somewhere right now.
Hi, doctor.
Hello.
No question, like, so the potassium permeability is following the ideal condition, like negative 90 millivolts. The sodium ion permeability is not following its like ideal condition, like it's